Today we will discuss about the B and T cell development. Okay, so as you all know that the B and T cell are the most important lymphocyte, which is which is very much essential for the adaptive based immunity. Okay, so how they develop and how they activate we will see in this class. So like other type of cells of the immune system, this is also formed from the hematopoietic cell. Okay, so this is the multipotent hematopoietic stem cell. Okay, so as you know that stem cells are the those stem means which is having the regeneration capacity, and it can be converted into any type of the cell based on the signal, what kind of signal it is getting. Okay, so it is the multipotent hematopoietic stem cell. Okay, so means it can create it is having the potential to produce many types of the lymphocyte or blood cells. Okay, so it will firstly divide into their progenitor cells. Okay, and then it will means further when it divide through the signal, it can create two type of cell. First one is the myeloid stem cell and another one is the lymphoid stem cell. Okay, and this myeloid stem cell is helpful to produce megakaryoblast, proerythroblast, myoblast and monoblast. Okay, and from here from the megakaryoblast it means indirectly it will give you the platelets. Okay, and from proerythroblast it will give you the erythrocyte or RBC. Okay, and blast it will give you the basophil, neutrophil or eosinophils. Okay, and then from the monoblast it will give you the monocyte, and from after monocyte it will give you the macrophages. Okay, so this all cell is essential for the innate immunity. Okay, so it is basically helpful for the innate immunity, and whereas this lymphoid stem cell which will give the lymphoblast as the progenitory cell, it then further divided into the natural killer cell, which could be part of the innate immune system as well as the adaptive immune system, and another one is the small lymphocyte. Okay, so this small lymphocyte or this is small lymphocyte progenitor cell further divided into either T lymphocyte or the B lymphocyte. Okay, if it is converted into the B lymphocyte, then finally it will give you the plasma cell or memory cells. Okay, and if this T lymphocyte will divide, then it will give you the either CD4 or humoral helper T cell, or it can give you the CD8 or cytotoxic T lymphocyte. Okay, so this whole process of synthesis of or the development of different type of cells is called as the hematopoiesis. Okay, this process is called as the hematopoiesis. Okay, and this all cell is produced from the hematopoietic stem cell. Okay, which is having the capacity to produce different type of red cells or the white blood cells, I can say. Okay, so these are the white blood cells. Okay, and here it is the red blood cell, and it is the part of the innate immune system, and this is the part of the adaptive immune system. Okay, so for development of this T and B cell, there are different organs in our body where the development take place. Okay, so the first one is the primary lymphoid organ. Okay, so what is the primary lymphoid organ? It is the central lymphoid organs. Okay, where the immature lymphocyte develops. Okay, firstly it will develop here, and then this in this organ the immature lymphocyte will differentiate, proliferate and mature to the mature to the immunocompetent cell. Okay, immunocompetent means still it is not encounter with any kind of the antigen. Okay, so this is happen in the primary lymphoid organ, and the example of primary lymphoid organ is the thymus and bone marrow. Okay, whereas the secondary lymphoid organ, where the mature lymphocyte will encounter with the antigen, and it means based on the antigen and encounter, it will effectively differentiate or proliferate into for the mass production. Okay, and it is the part of the adaptive immune system. Okay, and it include the spleen, lymph nodes, tonsils, appendix, and Peyer's patches. Okay, so these are the secondary lymphoid organ, where the mature lymphocyte will encounter with the antigen, and it will proliferate its number. Okay, from here, and it can initiate the adaptive immune response here. Okay, whereas this primary lymphoid organ is the initial stage, where the lymphocyte will develop firstly. Okay, and the positive and negative selection, all those things will happen here, and after that it will migrate to the secondary lymphoid organ, where it will encounter with the antigen, and what kind of an antigen it is encountering, based on that it will proliferate its number. Okay, that is called the clonal propagation. Okay, so what is the B cell? This B lymphocyte, named after their site of the origin in the bursa of fabricius in birds. Okay, or in the bone marrow in human. Okay, so these are the primary lymphoid organ. In case of the birds, it is present in the bursa fabrici, and in case of human, it is in the bone marrow. Okay, and it can produce the humoral immunity by the production of the immunoglobulin or the antibodies. Okay, and this cell belongs to the adaptive immunity. It is having the antigen specificity. This means it can also initiate the humoral immunity, and it is the how it produce the humoral immunity, by producing the plasma cell or antibody producing cell. Okay, so this is the B cell, and how it develop. So this is the flow chart of the development of B cell. So the differentiation of B cell occur in the bone marrow throughout of the life of an individual. Okay, so in case of the bird, it is bursa fabrici, and in case of the human, it is bone marrow. So it is firstly develop and mature. Both the things happen in the bone marrow in case of the B cell. Okay, so the first B cell lineage is the pro B cell, which is followed by the pre B cell, then immature B cell. Okay, the cells which express the IgM on its surface. Then finally it will produce the mature B cell. This cell will be having the IgM and IgMD on its surface. Okay, so this is the flow chart. So suppose this is the multi hematopoietic multipotential hematopoietic stem cell. Okay, from after that means further division, it will give you the lymphatic progenitor cell, and this lymphatic progenitor cell further means after certain kind of cytokine synthesis, it will give you the pro B cell. Okay, in the pro B cell, it is the earliest lineage of the B cell. Okay, here. Okay, and after that it will give you the pre B cell. Okay, in the pre B cell, here you can see that it is the IgM. Okay, IgM will synthesize on the surface of B cell. Okay, that is called the pre B cell, and after pre B cell, it will give you the immature B cell. Okay, in immature B cell, the IgM expression will be completed, and meanwhile the IgD expression will start. Okay, and then this is the final mature B cell, or we can say that naive B cell, because still the B cell is not encountered with the antigen. So that's why it is called as the naive B cell or the mature B cell. Okay, so finally when the IgD also will express on the surface of B cell, it is called as the mature B cell. Okay, so after producing the mature B cell, it will move to the periphery. Okay, periphery or the secondary lymphoid organ. This means this till this process it is called as the antigen independent process, because here no role of the antigen, okay, and it take place in the bone marrow, which is a primary lymphoid organ, okay. And after the generation of mature B cell, it will reach to the, means through blood stream it will reach to the periphery or to the secondary lymphoid organ, okay, which is antigen dependent, because here the mature B cell will encounter with the this B cell, and accordingly, like whatever signal it will achieve, it will divide on that basis, okay, because in this surface it is having the B cell receptor, okay. This B cell receptor is having, like in earlier classes we have seen that in the antigen, in the antibody, there is two type of chain, one is the heavy chain and another one is the light chain, right? So in that side antigen binding domain is present, and in this antigen binding domain it is highly variable, and so that's why this whatever antigen it will bind, based on that it will get the signal like I have to multiply to, means which should have the specificity towards this type of antigen, right? So it will multiply, means by dividing either plasma cell or either memory cell, okay. So this is the simple flow chart of the B cell development, okay. So this is the in the detail like how B cell development happens. So this is the pro B cell, okay. So from the lymphatic progenitor cell there will be pro B cell will synthesize, okay. And in pro B cell there is the immunoglobulin alpha beta heterodimer will form first, okay. So it will further give you the heavy chain, okay. So this is the marker, or we can suppose you are having multiple cells, so whatever marker is present on the cell surface, accordingly we can identify like this cell is present on which stage of the development, right? So these are the pro B cell markers like CD19, CD43, CD45R and CD24, as well as this immunoglobulin alpha and beta means now synthesizing, okay. It is just started here in case of the pro B cell, okay. Then the mu chain rearrangement take place, okay. So in case of the mu chain rearrangement means here the heavy chain, because in case of antibody class we have seen that the antibody name is given based on their heavy chain, right? So mu chain means it will give rise to the IgM antibody, okay. So that's why here the heavy chain rearrangement take place, and once it is take place it will give rise to you the pre B cell, okay. Other markers will remain same, only the this heavy chain means synthesis is completed, okay. And simultaneously the light chain synthesis is starting, okay. So in that stage it is called as the surrogated L chain, okay. Here you can see that in a larger view, this is the pro B cell where only the immunoglobulin alpha and immunoglobulin beta dimer is present, okay. And then here the heavy chain rearrangement take place, okay, which is a mu type of heavy chain, okay. It means it will give you the IgM antibody, okay. So after that means in pre-B cell the heavy chain rearrangement is completed and just started the light chain rearrangement, okay. So it is not completed yet, okay. So that's why it is present as a the light chain, this is the variable region, so that's why it is called as the B proteins, okay. And this is the heavy chain of the this is the constant region of the light chain, that is called as the lambda 5, okay. And the whole this light chain is called as the surrogated light chain because it is still not completed, okay. So that's why it is called as the surrogated light chain, okay. then it will converted into the immature B cell. Okay, because now what will happen is light chain rearrangement will take place. Okay, so now this in when the immature B cell will produce, that time the heavy chain as well as the light chain rearrangement will be completed, and it can be means synthesized as an intact molecule of IgM. Okay, so firstly in immature B cell there will be first antibody which synthesized, that is the IgM. Okay, means we know that in the B cell there are two type of antibody. First one is the IgM and another one is the IgD. Okay, in the previous class we have read that. Okay, so there is IgM and IgM and IgD, right? So firstly IgM will produce, and once IgM is produced, it is called as the immature B cell, because still the IgD is not produced. Okay, so here you can see that in the mature cell there is two type of antibody. One is the IgM, okay, and another one is the IgD. Okay, so then it is called as the mature B cell. Okay, so when I am talking about like rearrangement, then what is happening there? So here you can see that in the antibody there is two chain, heavy chain and light chain. Okay, this is the heavy chain and this is the light chain. Okay, and this heavy chain and light chain is made up of different type of gene, that is called as the variable diversity joining gene. Okay, VDG. Okay, VDJ. Okay, so that is called as the VDJ recombination, and because of that only it is having the high variation to bind with different kind of the antigen. Okay, because of these genes. Okay, so here you can see that in the heavy chain there is three type of combination, VDJ. Okay, and whereas this in case of light chain there is the only two type of combination, one is the V and another one is the J. Okay, so while when we talk about like heavy chain rearrangement, that time this VDJ recombination will happen, and the intron part will remove there is different locus from V7 to J32, like that. Okay, so there are multiple combination. Okay, it will occur, and each B cell is having a different type of recombination, right? Because it is a kind of random recombination. So based on that it is this rearrangement will take place. In case of the heavy chain there is three type of combination, V, D, J, and in case of light chain it is the V and J only. Okay, two type of combination is there. Okay, so in case of pro B cell, firstly the immunoglobulin alpha and beta dimer will be present, and then when it converted into pre B cell, their heavy chain through this duration heavy chain rearrangement will take place, and in pre B cell there will be the heavy chain will be present, and meanwhile the light chain rearrangement will start take place, and once it converted into the immature cell, the intact IgM molecule will be present on the surface of B cell, and once it is converted into the mature B cell, it will be having both the antibody IgD as well as the IgM. Okay, so and after once this mature B cell is produced in the bone marrow, which is the antigen independent phase, it will migrate through the blood stream, it will migrate to the secondary lymphoid organ, that is the lymph node. Okay, and here this lymph node it will encounter with the antigen. Okay, and based on this antigen encounter, means what kind of antigen it is, it will multiply particular variation of B cell. Okay, so here it will firstly divide, some B cell will divide into plasma cell and some will divide into memory cell. Okay, which is having the same specificity for this antigen. Okay, and they will multiply to increase their number, and they will neutralize the antigen. Okay, so this by secreting the particular kind of antibodies. Okay, so this is the development of B cell. Okay, so there is two phases. One is the antigen and independent, and another one is the antigen dependent. Antigen independent phase always take place in the primary lymphoid organ, and the antigen dependent phase will take place in the secondary lymphoid organ, that is the lymph node. Okay, so in the primary lymph node or in the bone marrow, firstly the B cell will converted into the the lymphotic progenitor cell will converted into the pro B cell. Okay, in the pro B cell there will be immunoglobulin alpha and beta dimer will present. Okay, apart from this CD markers, CD45R, CD43, CD19 and CD24. Okay, and after certain time, like after getting the signal from pro B cell to pre B cell conversion, there is the heavy chain rearrangement will take place. Okay, in the heavy chain rearrangement means this VDJ recombination will take place. Okay, means which kind of combination should be there. It can be like V7D 8J8 like that. Okay, so there is I don't remember the exact number of the like how many V is there, D and J is number, the number is I don't remember, but it is like that. Okay, so it is the random process, like this V can recombinant with either one of this D or one of this J. Okay, so each B cell is having the equal chances to recombinant with other type of D and J, and vice versa like that. Okay, so this type of rearrangement take place here. Okay, while transition of pro B cell to pre B cell. Okay, once this pre B cell is formed, it is having the intact heavy chain, and it is having the surrogated light chain, because this light chain synthesis is yet completed. Okay, so once this pre B cell receptor is converted into immature cell, that time the light chain synthesis is also completed. Okay, and the IgM molecule will synthesize on the surface of B cell. Okay, and after that it will divide as a mature B cell, where the IgD antibody will also there. Okay, so this is the development of B cell in the bone marrow, and once this mature B cell is produced in the bone marrow, it will for their effectory function, it will through blood stream it will migrate to the lymph node. Okay, and in the lymph node it will encounter with the antigen, and after encounter with the antigen it will divide either in the some part of the this B cell will divide into plasma cell, and some part will divide into memory cell. Okay, and this plasma cell will give you the secreted antibody which is specific to this antigen, and it will neutralize this antigen to reduce the infection or to eradicate the infection. Okay, so this is the B cell development. Now the development of T cell we will see. So in case of T cell, this T cell is originated from the hematopoietic stem cell in the bone marrow, and undergo positive and negative selection in the thymus to mature. Okay, so unlike the B cell, like in case of B cell, both the thing, the development as well as the maturation take place in the bone marrow itself. But in case of the T cell, it is firstly develop in the bone marrow, and for the maturation it will go to the thymus. Okay, so that you have to remember. Okay, so the maturation of T cell take place in the thymus, whereas the in case of B cell, both the things happen in the bone marrow itself. Okay, so in case of T cell, what is happening is this is the common lymphoid progenitor cell. Okay, this is the progenitor cell, and firstly it will divide into the DN1 cell. DN1 cell is nothing but it is the double negative cell. Okay, double negative cell means it is not having the CD3 surface marker, CD4 surface marker, or CD8 surface marker. Okay, so this is after that it will converted into the DN2, that is the double negative 2. Okay, here what will happen is the RAG1 and RAG2 gene will take place. Okay, enzyme, these are two enzymes which will take place. Okay, and it will like here you can see that this is the T cell receptor. Ultimately we require the TCR or the T cell receptor. Like in case of the B cell, there is the BCR, B cell receptor. Okay, what is the B cell receptor? This IgM and IgD is only the B cell receptor. Okay, because it is going to bind with the antigen. Okay, so the like that here the T cell receptor which is going to bind with the antigen. So here it is made up of two type of chain. One is the alpha chain, another one is the beta chain. So like in case of the antibody, the light chain is made up of the V and J segment. Okay, so here also the alpha chain is made up of the V and J segment, whereas this beta chain is made of the VDJ segments. Okay, so here also this rearrangement is happened due to the presence of this RAG genes. Okay, so these are the rearrangement genes. Okay, it will help to rearrange the this alpha and beta chain. Okay, so then DN1 will converted into the DN2, which is the double negative. Okay, and then this double negative 2 will converted into the double negative 3. Okay, here what will happen, the D and J segment will is spliced. Okay, and only the V segment will be there. Okay, and in the conversion of DN3 to DN4 there will be VDJ combined with constant region. Okay, now the constant region will form here in case of the DN4. Okay, this is the DN4 where you can see that CD3 is present. Okay, means it is going to start the synthesis of CD3. Okay, here, and there is the beta chain and alpha chain is now present here. Okay, this is the starting of the TCR. Okay, so after cell division, what it will give you the double positive cell. Double positive cells means it is having the receptor for that CD8 as well as the CD4. Okay, and then again it will rearrange the alpha chain. In case of the like a double positive cell, it will rearrange the alpha chain as well as the beta chain, and then it will go for the positive selection. Okay, positive selection means it will firstly those CD8 and CD4 cells will and which can bind with the MSC molecule, because you know that T cell only encounter the antigen with the help of MSC molecule. Okay, so once this T cell is able to bind with the MSC molecule, those T cell will be selected. This type of selection is called as the positive selection. In the next part, in the negative selection, what will happen is suppose this is the T cells. Okay, most of the 90 to 95% T cell will not able to bind with the MSC molecule, so they will go for the apoptosis, and those type of T cell will die. Okay, and rest of the T cell which will able to bind with the MSC molecule, this type of selection is called as the positive selection. And after positive selection, it will go for the negative selection. In case of negative selection, what will happen is those T cell which is having the higher affinity for the self antigen, that will also go for the apoptosis. Okay, so these cell also go for the apoptosis, which is having the higher affinity for self antigen. Okay, so this is called as the negative cell. Okay, negative selection, and after that only 2 to 4% cell will remain which is having the desired affinity for the foreign antigen. Okay, so it will go to the like secondary lymphoid organ, and it will then it will encounter with the particular type of antigen, and then it will neutralize those antigen. Okay, so what is happening here in case of the development of T cell? Firstly, the common lymphoid progenitor cell will converted into the DN1 cell, that is the double negative cell. Okay, in case of double negative cell, there is no cell surface marker, which is this CD3, CD4, CD8 are the T cell surface marker. If this surface marker is present on any kind of cell, then you can identify like that this cell is T cell. Okay, so CD3 is one of the means important or specific cell surface marker for T cell. Okay, so that is absent in case of the dn1 to dn4. Okay, so in case of dn1 from means when it is transition is happen between dn1 to dn2, that time what will happen is rag1 rag2 gene will start the rearrangement of this alpha chain and beta chain. Okay, then in case of dn2, what is happening is dj segment rearrangement is happen. Here you can see that there is vdj. Okay, so firstly the beta chain rearrangement is happen, like their heavy chain rearrangement take place first, so here the beta chain rearrangement will take place first. Okay, so here what is happening is it will splice the V chain and then combine the D and J segment. Okay, and after combining the DJ segment, now it will combine the VDJ segment in case of the DN3. Okay, then finally it will make the constant region of beta chain. Okay, it is here the beta chain will form as well as the CD3 marker will also present. Okay, and after certain cell division, it converted into the double positive cell, which is having this beta chain as well as the CD4, CD8 and CD3 marker. Okay, that's why it is called as the double positive, because it is having CD4 as well as the CD8 cell. Okay, and then it will rearrange the alpha chain also, and it will now mature or naive T cell. Okay, and then after positive and negative selection, what it will do is it is having both type of receptor CD4 as well as the CD8. Okay, but some of the T cell, like in case of B cell, some of the B cell will firstly divide into plasma cell, and some of will divide into memory cell. Like that, here also T cell will, some of the T cell will divide into specific CD4 T cell, and some of will divide into specific CD8 T cell. Okay, by down regulating the other T cell receptor. Okay, because it is having two types of the T cell receptor. So if it is dividing into the CD4 T cell receptor, then it will down regulate the its CD8 receptor. Likewise, in case of CD8 T cell, it will down regulate the CD4 T cell receptor. Okay, so it will happen after the positive and negative selection. Okay, so this positive, in case of the positive selection, what will happen is those it will take place in the thymus cortex. Okay, that also you have to remember, like which type of selection place where. Okay, so the positive selection place in the thymus of the cortex of the thymus. Okay, so in the cortex of the thymus, what is happening is the population of T cell or the naive T cell, what it will do is it will here it will check for the, like it can be able to bind with the MSC molecule or not. Okay, so suppose some of T cell will not able to bind with the MSC molecule, those type of T cell will go for apoptosis, and those type of T cell will die. Okay, and rest of the T cell will migrate to the medulla of thymus. Okay, where the negative selection take place. Okay, in the negative selection, what will happen is, in the negative selection, the positive T cell which is able to bind with the MSC molecule now check for that the ability, like it is not showing higher affinity for the self antigen. If it is showing the higher affinity for self antigen, then it will go for the apoptosis, and rest which is not showing higher affinity for the self antigen, that will migrate to the secondary lymphoid organ or the lymph node. Okay, where based on the different type of signal, it can converted into the, like in case of the CD4 or the helper T cell, it can converted into the Th1 type of cell, Th2 type of cell, Th17 type of cell, and T regulatory cell, as well as this CD8 can go and perform its function as a cytotoxic T cell. Okay, understood? This Treg cell is important to balance these two type of cell. If suppose there is more CD4 and CD8 cell, then there is a chance of higher cytotoxic mediated immunity, which can cause the autoimmune disorder. Okay, because the means more immunity is also not very good. So to regulate those type of cell, there is Treg which can inhibit the it will give the feedback inhibition to CD4 and CD8 division, so that it can't be further divided into more CD4 and CD8. Okay, the amount is necessary, that much amount will only form produced. Okay, so this is the development of T cell. Understood? So this is the brief of both the cell development. So this is the hematopoietic stem cell, then it will converted into the lymphoid progenitor cell, and if it is differentiating into B cell, then firstly the heavy chain immunoglobulin heavy chain rearrangement will take place. Okay, and it is called the pre-BCR. Okay, where here you can see that surrogated light chain is also started to produce, and if it is not produced, then it will go for the apoptosis. Okay, and once this BCR is converted into the BCR or the immature BCR, okay, that time IgL or immunoglobulin light chain rearrangement will take place. Once this synthesized completely, it will converted into mature cell, where the IgD will be also produced. Okay, so that is the mature B cell. Okay, this is the mature B cell, and this is the differentiation to T cell. Okay, so firstly the beta chain gene rearrangement will take place, like in case of immunoglobulin, like IgM heavy chain rearrangement take place. Here beta chain gene rearrangement will take place firstly. Okay, then in pre TCR there is beta chain present, and CD3 is also present. Okay, if this type of signal it will not get, it will go for the apoptosis. Okay, then finally when it converted into the mature T cell, that time it will regenerate the alpha gene rearrangement will take place. Okay, and here in the double positive case, there will be TCR as well as the CD3 CD4 and CD8. This is the CD4 and CD8 cell surface marker will be also there. Okay, and in the thymus, it either some of the T cell will divide into helper T cell, and some of them will divide into killer T cell. Okay, or the cytotoxic T cell. Okay, so this is the whole process of the development of T as well as the B cells. So regarding this subject, one question has been asked: B lymphocyte originated from the bone marrow, whereas T lymphocyte originated from thymus, bone marrow, spleen, liver. What will be the correct answer? So yes, the correct answer will be bone marrow, because as we discussed, like B lymphocyte originated as well as mature in the bone marrow, but T lymphocyte originated in the bone marrow, but maturation will take place in the thymus. Okay, so that's why the bone marrow is the correct answer. Okay, whereas thymus is the place where the T lymphocyte maturation take place. Okay, whereas this bone marrow is the place where T lymphocyte originated from the hematopoietic stem cell. Okay, so the correct answer of this question will be bone marrow.